So I want to continue where I stopped last time, and this was about hair cell and hair cell models. And there I stopped here, which is introducing a bit more advanced or revised, they call it revised hair cell model, which splits the processing into the processing of the calcium ions, potassium ions in the hair cell. That's quite similar to what we saw before. Yes, controlled by the displacement of the stereocilia, and then the throughput through the channel is controlled by the displacement. Then we have some time constant for ions going through the membrane and some generator, and so this would be again similar to what we saw before for the dynamics. And then there is more or less just a model for the probability of action potential at the second part. So this is the membrane potential. This is a passive model, just controlled by this resistor, which is controlled by the displacement of the stereocilia, and then the rest is passive, except for the energy sources here, of course. And then what happens here in this part is then a separate model for the probability, so that they split it more or less, but it's not too different from the previous model, where we also had this nonlinearity in this in the control of the permeability of the throughput of the potassium ions by the displacement of the stereocilia. So this was more or less integrated into that, and here there's a separate model about that. And continuing from there, we have some, maybe I should point with this, we have some neural activities on the nerve fibers of the auditory nerve. And then I want to show a little bit what processing comes then on the signals which are transmitted by the auditory nerve. And here we see a little bit, let's say, an overview of what happens. Here we see kind of symbolized the cochlea of the, depending on if you look from the right or the left side, so of the two ears. Then we have the auditory nerve, and this is connected to the CN, stands for cochlear nucleus. So this is the first part which receives main information by the auditory nerve and does the first steps of processing. And then we have some different regions in the brain which further process, and you see they are connected quite in a verified way, so in a very neat way, with the different regions connected to each other. But mainly the information flow goes from bottom to top in this graph. And so this is the signals which are received by the cochlea, and not the other way around, like the things which could control something by these efferent nerve fibers are not shown here. Here, this is more what happens to the actual audio signal and its representation on the auditory nerve. So it's processed in these stages. The abbreviations are explained here. I don't need to name all of them. And then finally, let's say the most semantic processing is then on the auditory cortex. And if you see that the main information flow goes more or less across from this side, let's say from the left ear to the right side, it really in location in the body. So it goes more from the left to the right and from the right to the left. So these are the main information flows, but you see there are some interconnections between the two sides, so that some joint processing can take place, which is important for localization of sound, as you can imagine, what we had in the first lecture. And so the first processing in the cochlear nucleus, there's a bit more of what is investigated, what kind of cells are there to do a first rough, let's say, audio processing in this stage. And I can show you a little bit of that. And this then shows some kinds of different cells which are in the cochlear nucleus. You have different shapes of cells, as you can see here, the names explaining just the shapes, more or less spherical, bushy cell, stellate, like star shaped, and so on. And what is shown associated to the different shapes of the cells is, as far as it has some specific properties have been found, they are shown by, let's say, their spikes and duration, the connection between spike rate and duration. It means if a sound is switched on at time 0, then there would be some, like here, some peak, some overshoot of the firing rate, and then it would go down. And you see that there are some which mainly produce a large spike rate for a very short time, and then there's no more activity. So this would be onset cells, which signal that there's a sound onset somewhere. Yeah, so and so you can see that there are some different kinds of behaviors in terms of time. There's a so-called pauser, like which has first a spike or peak after the onset, and then there's a pause, and then there's increased activity after a certain delay. And so you can imagine that there's already some temporal processing done here, which helps then the next stages in auditory perception to get more semantics out. But you still have to know that, of course, there are different ranges for different frequency ranges, because we have the hair cells placed on the basilar membrane, which are at specific characteristic frequency positions. And so the nerve fibers would be processed also to some extent separately, but to some extent jointly, so that there'll still be some dependency on the frequency content and some dependency on, let's say, the temporal behavior of some sounds which are coming in. And then, but the frequency would still be represented by having different, of course, always multiples of those cells, and each would be responsible for certain frequencies or certain frequency regions. And that's what makes then enables the next stages of the processing to get some meaning out of this firing rates or spike rates which we have here. So, and there is some neural processing in the cochlear nucleus. There's some subpart of it. It's called anteroventral cochlear nucleus. There are mainly these spherical and globular bushy cells, and there's a relative small number of large cells which can have strong excitation. That means they can have high firing rates, and they have more, this is called primary-like responses. So that means they resemble the primary firing rates and distribute that further. So their firing rates are quite similar to those of the auditory nerve fibers. So they would more or less distribute the neural activity Then we have these stellate cells, or they are also called chopper cells, which obtain inputs from several auditory nerve fibers. That means some frequency ranges, and also from other types of neurons. And they specifically respond to pure tone stimuli with regular rhythmic bursts, with burst frequencies which are not related to the stimuli themselves. But they have a narrow frequency tuning. This would be for frequency or pitch recognition, and they have a large dynamic range. So just to show a little bit what is known about some of the cells which are processing the sound. And there's another part of the cochlear nucleus, the posterior ventral part. And they are mostly these onset cells, which are these onset cells, for example, react to pure tone bursts with just a single action potential, or to inputs. It would also process inputs from many auditory nerve fibers, because onsets normally, if you have an impulse-like signal, this would have a wide spectrum, and so this would also be covered by the fact that many auditory nerve fibers are processed together in these onset cells. But they have some response latency jitter to the onset, which are in tens of microseconds. So there is some variation how quickly they react, but still in fairly short time in terms of just processing the envelopes of the sound, not the sound itself. But so they can also mark beats in complex sounds. And then there's another third part of the cochlear nucleus, the dorsal part, and there are these pyramidal cells amongst other types, but they are complex pauser type temporal response patterns. As I showed you before, they would have first a peak, and then they would have kind of a pause before they again show higher activity. And they can be inhibited by some frequencies and excited by other frequencies. So that means by certain fibers, they would be activated, and other fibers could inhibit the activity. And so this could be, they can be used for detection of spectral contrast. If one cell is excited by certain nerve fibers coming from a certain characteristic frequency position, and other fibers which are in the neighborhood would rather inhibit that, they would then have a stronger reaction on tonal signals, where there's just one large activity at one position and lower activity in the neighborhood, for example. So that's why they could be able to detect spectral contrasts. Yeah, so that is a little bit of introduction of the neural processing, but since on from there, it's very difficult to actually build models out of that, except for just, let's say, we call it psychoacoustic experiments, or also other psychological or so experiments, by knowing what's going on for the understanding of sound and the further interpretation of sound. This could be done, of course, with neural networks, but then on a global level. This would not try to model the individual components, but just get some input sound and process and produce some reaction, which would then give some information about how humans would perceive that. So, but that's the point where normally the specific modeling is, there's no specific modeling of these neural processings which are happening here. And further things are then, let's say, modeled more according to what I said before, psychoacoustic experiments. And this would be especially the case for what I will explain in the next section, which is about masking effects. And the data comes from psychoacoustic experiments, which I will explain again. I already used masking a little bit already for explaining the frequency resolution of the auditory system, but there are more specific masking effects, and I will show you that in the next part. Are there questions regarding this part of the neural processing?
I have a doubt. The frequency selectivity of the fibers, right? Is it related to the shape of the nerve?
It's since maybe go back to the or maybe come back. So we have these pyramidal cells, and are they shown? Those are these fusiform, these are the pyramidal cells. And you see they have multiple connections, and they could be connected to multiple, let's say, to multiple auditory nerve fibers, which might come from different positions on the basilar membrane, maybe neighboring positions. And some of the input which they get here would inhibit the activity, and other would excite some activity. And that's why the combination of excitation and inhibition that makes in the end the activity. And that's why this can emphasize spectral contrast. Let's say if the inhibition is as strong as the excitation, there might be no activity. That means if there would be kind of a flat spectrum, there would be not so much activity as if there would be one specific tone and nothing in the neighborhood of the, let's say, of the neighboring hair cells. There would no activity being arriving at the inhibition part, let's say. So that's how you can imagine how this works to emphasize some spectral contrasts by certain cells which have this behavior.